Good morning, everyone. I welcome you all to this 18th day of CSIR Life Science question and answer discussion session. Past 3 days, I was met some technical issue in regarding YouTube, so I was not able to post the videos today on of this 13 days training. Okay, discussed about pa past 17 days we are discussed about 5 part C questions and all the unit part B questions. Okay, in this session we are going to discuss about methods in biology. Okay, what are the methods in biology? Methods means it includes the microbiology methods, molecular biology methods, genetic engineering methods, and protein engineering. All those things are comes under the method category. Specifically, as I am said in earlier days of class, CSIR examination is the research-based question in the exam point of view. In the exam, the many of the questions are relevant to the research field. So, research means that is having n number of experiments. No, if for example, if you are going to facing any microscopic related study questions means first of all we want to know the structure and what is the principles behind the microscope. So every methods having their own principle. So in this while you are studying this 13th unit methods in biology, you want to understand, you want to get clarification about the methods, which are the methods involved in the microbiology, which are the methods in the immunology, methods in protein engineering, and their principles is very important. Just to google it in the syllabus, 13th unit syllabus, you can find the various topics. Okay, just type the topic name and and add it principles. Okay, for example, light microscopy, so just google it with the light microscopy principle. So you can get more understanding about the concept. That is the one thing in the this methods in biology studying. That is all about the 13th unit. Okay, next we will enter into this session and further I will discuss. Okay, first question is all about the microscope. Most important property of any microscope is it is resolution. Resolution is denoted as the D. Can be calculated from the formula D equal to 6.1 lambda n sine alpha. This is the statement. Here, the D is distance between two distinguishable objects. Alpha is the wavelength of the instant light. Okay, alpha is the wavelength of instant light. Sorry, lambda. Alpha is the angular aperture. Alpha is the angular aperture. N is the refractive index of medium. These are all about the equation. First, they provide the equation is respect to the resolution, and then they are explained the terms. Okay, what they asked. Given below are several suggestions to improve the resolution of microscope. Here they provided the suggestions to improve the microscope resolution. As per their study, they mentioned that there are many methods are available to improve the microscope resolution. Here they are mentioned some of two methods. What is the methods? Decrease the wavelength of instant light. While you are decreasing the wavelength of instant light, you can improve the resolution. Second option, using the oil which has higher refractive index. This very important thing about this microscopic concept. If the microbiology people, botany people, all of them may know, the biotechnology people know, while you are seeing the organism under the 100x, we are using the this kind of higher refractive index having uh, like oil in the slides. This higher refractive index having the oils are shown a better resolution, shown better image clarity. it's very important thing. So two things are here: decreasing the resolution of decreasing the wavelength of instant light. Another thing is the higher refractive index oil. This higher refractive index means this material with a higher particularly that that material, okay, the higher refractive index having material shown refractive index angle, okay, the angle of refraction will be smaller than the angle of the incidence. This higher refractive index oil, which having the that known in, we all know that emulsion oil, that emulsion oil material, it's a high dense and viscous material. This higher refractive index of material that like some oil, vegetable oils having the nearly 1.4 refractive index, and cedar that is one kind of emulsion oil that having 1.5 refractive index, that is a higher refractive index in the current research areas. Okay, so this refractive index principle is that refractive index material that shown the angle of refraction is smaller than the angle of the instant. This is the very important thing about the refractive index. So it's shown the higher refractive the oil which having the higher refractive index will improve the image resolution in the microscope. Okay, these are all things. Now we can ask, what is the relation between that statement and the question? There is no that here you can see the lambda value, the wavelength of instant light. So while you are changing the this lambda instant light and n value, n is the major thing. For example, emulsion oil's refractive index is higher, means that resolution become increase, that shown a better result, which look it good, it gives a better image. So this refractive index, while using the n is the refractive index. The dimension oil refractive is 1.5. This playing a vital role in the multiplication and division part. Okay, next thing is the question. It's a direct question. It's a sum based question, but it's the easiest one. In this question, the mean and standard deviation of serum cholesterol is population is in the senior citizens are assumed to be 200. So mean is a 200. Standard deviation is 24 mg per ml. As I said in our part A discussion session, the probability and statistical questions are most widely asked in the CSIR examination. So you want to understand, you want to get better idea about the statistics and probability. In a random sample, as I hear they mention, the mean is 200, standard deviation is 24 mg per TL. The random sample of 36 senior citizens, okay, they had taken the 36 senior citizens, what value of cholesterol should lead to rejection of null hypothesis? Rejection of null hypothesis. What does the rejection of null hypothesis means? Rejection of null hypothesis is the okay, right, this is the answer, this we want to get. Well, null hypothesis means, in this null hypothesis, that probability, okay, that the probability is shown the it's no difference between certain characters of the population, that is the exam, that is the denotion. Okay, they provided the 36 senior citizen, so that in that population no difference in the particular term, that is the cholesterol variation, no difference, all of them getting a same. What is the here there as a rejection? How to avoid this rejection of null hypothesis? First, we want to understand what is null hypothesis. Null hypothesis means that is no difference between the certain characters of, okay, character here is the cholesterol. So no difference in the cholesterol between the all 36. This is the null hypothesis. All of them having same value. Here the question is how, which is lead to rejection of null hypothesis. Rejection of null hypothesis means all of them having a different, all of them having a different cholesterol value at 95% confident level. What is 95% confident level? This 95% of confident level means this is the a particular region which don't having the specified values. So here you can see this as per their equation, the calculation, they get the value is the 8. Okay, the number of values 8. So the rejection of null hypothesis is between that is below 192 and it's about 208. This is all about the standard deviation. This the mean value is the mean value which having the 200. They are getting the differentiation value is 8. After this calculation they receiving the rejection of null hypothesis at 95% level is below 192 and above the 208. I will send the model examples in your Telegram group. I'm having examples for this question. We will discuss. I will send it in the group. Okay, next part of the question is very interesting and easiest question. In response to a drug, change in protein level were examined in a cell line. Okay, they are providing a drug, their change in protein level is examined in the cell line. A chase experiment. The plus chase experiment was performed in the performed using the S35 labeled methionine. Here the denotion, the statement, plus chase experiment, they are using the using with the S35 labeling. This what is the plus chase experiment? Okay, just first cells were grown in the medium. After that it's exposed to the particular radiation. That is the plus chase experiment. This plus chase experiment, many of the cellular processing, okay, the cellular activities are understandable by this plus chase analysis. Processing, it's understandable while using this plus chase experiment. It's kind of electrophoresis method. It's kind of experimental with they are growing the cell in a medium. After the particular period they are exposing the cells to the radiation. So the radiation molecule is the S35 labeled methionine. They are added to the cell. This is the statement about the experiment. A comparison to untreated sample, the following observations were made. Few minutes after the simulation, protein X accumulated after the particular period of the radioactive label exposure. Protein X accumulated. That was followed by reduction in the protein Y and protein Z. So two things are the first accumulation of X, after that reduction of Y and Z. The correct interpretation of this observation would be option is that D. Information is not sufficient to distinguish between the three possibilities stated above. Okay, here the mention they are given the statement. First one is statement is a experiment. That experiment they are what they mention, comparison to untreated sample. Okay, they are only explain about the untreated sample. This untreated samples are following up happen few minutes after simulation. That is the simulation means here the radioactive labeling, the radioactive molecule cells are undergone the radioactive molecule exposure. Protein accumulated and this followed by reduction in the protein Y and B. This is the statement. What is the correct interpretation? A is the protein, X is the protease. and degrades Y and Z. Okay, what is here this statement explain? They consider, imagine X is a protease enzyme. So this X protein is accumulated, protein X is a protease, so it's accumulated in particular area and degrade the Y and Z. This is the one statement. It may happen. After that, protein X is transcriptional processor that control the expression of Y and Z. So its reduction happen. Another option, the expression of protein X and loss of Y and Z unrelated. No relation between this first statement and the second statement. The protein is accumulated and the reduction in the Y. These are all not, and that is not related. These three things are not the correct answers. We can't able to select the correct one, so they are mentioned. This is the not sufficient data, we need another data. What is the another data? And comparison between untreated samples. Why we want to mention the untreated sample? While they are for keyword, one keyword here is we untreated samples are similar, they are not vary. Whenever you are going to do experiment, we need treated and untreated. Here they so we want to do comparison between treated and untreated, but here they what did, they did with only untreated cells. So it's wrong one. So this last statement, that is the not relevant statement. Okay, next about, okay, a gene from genomic library. This is all about the genetic material. A gene from genomic library is screened by using hybridization technique. The hybridization technique. After hybridizing the probe, usually a stringent washing step is given. After the hybridizing the probe, usually a stringent washing step is given. The following statements are given to explain the stringent wash step. What is the stringent wash step? This is the statement. Here they ask, finally, the what which combination of above statement is most appropriate to stringent washing step? First of all, we want to understand this hybridization technique. Okay, in this hybridization technique, we all know this normal genetic engineering method and the molecular biological studies all having this kind of method. This hybridization technique, the single strand DNA and RNA molecules that are annealed with the complementary DNA or complementary RNA to it's lowering value, they are lowering the temperature in the particular environment that single strand molecules are annealed with that cDNA molecule. Okay, the cDNA and single strand RNA or DNA that are complicated, that are complexed with the cDNA or cRNA. Okay, they are making the library compound, they will helpful for the sequencing. When it happen, while they are decreasing the temperature of the particular solution medium, that are comes to annealing. Okay, annealing means binding of cDNA and the that single stranded DNA happen with annealed with particular low temperature. This is one concept. Here they are using the hybridization probe and stringent step. Second thing, we want to understand the hybridization probe. In this hybridization probe, latest research areas, the researchers are using various hybridization probe, that is usually from 100 to 1000 base pairs. 10,000 base pairs also they are available. In this this DNA probes are uh, having particular specific library sequences, that is the highlighted molecules, that is helpful for the make a library. So length of the particular hybridization probe should be in the 100 to 10,000 base pair. In this stringent step, uh, okay, stringent washing step. This stringent washing step means they are during the hybridization, the labels are okay. The labels are added extensively. Okay, extensively means after the binding, some of the molecules are they attached over the DNA. That is extra molecule. That This stringency, okay, stringency, that is denoted the degree of homology between the probe and the filtered bound nucleic acid. That is higher the stringency, okay. That is exactly bind to the cDNA, that single stranded DNA which exactly bound to the cDNA. Some of times that alternative strand, yeah, that alternative nucleotides are not available, so means that there is no stringency is available. This stringency washing step, they are providing the some particular amount of temperature, okay. They are higher temperature, low salt condition. Each in every step of the hybridization, the temperature is very highly influenced in the highly influenced. While you are using this kind of stringency washing is in the step, they are using the high temperature and low salt. Very very important thing, the high temperature low salt. What is the stringent washing step means? That is high temperature and low salt. Okay, in this washing step they are using the saline sodium citrate. Okay, they are using the saline sodium citrate as a salt. They are adding the low concentration of saline sodium citrate with high temperature. In this temperature, this on extra that unextended steps are removed. Okay, remove the excess probes is the basic step. So this low salt concentration and the high temperature unbound nucleotides. Here the next, now we will enter into the appropriate washing step. Washing step is a 1, 3, 4 is a correcting. Take care of removing unincorporated and non-specifically hybridized probe. Okay, now you got the point. The unincorporated and unspecified hybridized probe. Second, stringent washing done, solution having low salt concentration, keeping high temperature. Salt present in washing solutions support hybridization to stay intact by shielding the interface of water molecule. This step, this salts helpful for the their interface, shielding the interface of the water molecules. These three things are important. Okay, next question. Small small molecular weight compound affect the activity of luciferase differently. This is the statement. Small molecular weight compounds affect the activity of luciferase enzyme. It's a one enzyme which playing in this vital role. The oil or water solubility of various compound is one property important of its effect on luciferase. Water or oil solubility means the non-polar compounds are oil. Polar compounds are water solubles. The straight line of the graph was obtained by plotting the activity data with 50 different compounds. The luciferase activity of two new derivatives of benzene. Here, the note the word uh, benzene. Okay, new two derivatives of benzene. Benzenes two, they are having a two derivatives means they are adding some extra compounds, extra molecules, extra atoms to the functional group of benzene. Derivatives means the addition of the compounds, molecules, atoms at the R group. the R group of the to the benzene. These are the functional groups. Any atom, any molecule that can add it to the functional group A region, that is the R region of benzene. Okay, that benzene structure you all know. So somewhat that bind at the any position. That is the A and B, two compounds. Here, the oil or water partitioning coefficient is given in x-axis, y-axis luciferase activity. A having higher activity, B having lower activity. The polarity, just imagine here they mention which of the following statement is correct. Okay, here they are adding the B is one here, right? A is methyl and B is amine. Just remember this. A is methyl, B is amine. Okay. Now we will go to this question. As per the statement, while methyl is added to the benzene, that become a non-polar. That is the bond sharing between the both methyl and benzene. Sorry, non-polar. And while they are adding amine, okay, that is sorry, that methyl, while you are adding the methyl, that polarity is become polar. That benzene, it's comes to polar region. Okay, benzene rings are the hydrophobic, that is the non-polar compound. While you are adding these that methyl group to benzene, that benzene polarity become reduced, and that is comes to non-polar. That is comes to the polar region. That their polarity index is 0 to 9. 0 means non-polar, that is the benzene kind of chemicals. While you are adding the CH3 methyl group to the benzene, the polarity become increase. Polarity increase means that may comes to 4 or 5. Polarity index, normal standard index is 0 to 9. The polarity of the benzene is 0. Well, they are adding the methyl group, the polarity of the that particular compound molecule, the benzene molecule, is become high. That may comes to 5 or 4. So the polarity index is here 5 or 4. While you are adding the amine group, that is the amine group is the, we are all know amino acids having this amine group gives the their non-polar effect. So where you are adding the amine to benzene, that molecule comes also in the state of the non-polar nature. Non-polar nature means that is the comes to 0. So here the B molecule is 0, that is amine added benzene. B is amine added benzene. A is the methyl added benzene. Methyl added benzene having polarity index high index, this having the low index. So this by the statement, the non-polar compounds, okay, the non-polar compounds having a low inhibition activity, low luciferase inhibition activity. The non-polar compounds having low luciferase inhibition activity, but the polar compound shown higher inhibition activity. Okay, next concept in the this question, the researchers was repeating a facts. We all know may about this. You all know about this facts. Facts means flow cytometric analysis. In this flow cytometric analysis, this is very important in the cancer biology research or any cell based research. This flow cytometry analysis, that fundamental process is the signal, individual cell signal receiving. In this experiment, they are repeating the FACS experiment, somehow got confused with the labeling of tube. Okay, they got confusion with the labeling of tube. There are 4 tubes one control. So one control and C tube with no fluorescent label, one stand, one S1 that is labeled with FITC. That FITC is one kind of label used in the this flow cytometry. This FITC is most widely used this compound in this kind of experiment. Okay, that fluorescein iso isothiocyanate. The fluorescein isothiocyanate. This FITC is the fluorescent thiocianates. Fluorescein isothiocyanates. FITC. Another is one stand, that is 2. Another stand is the S2 with the PE label. PE is propedium. I think so. Propedium. Wait. Yes, right. With having they are likewise they are adding various kind of dyes. Okay, this kind of experiments, all of them are very very sensitive. Okay, and this staining of all these stainings, PE cyanate 7, PE cyanate 2, all of them are they are providing the color fluorescence. Okay, FITC, PE, where various colors are produced from this kind of dyes. Okay, these colors are recognized by the system, and the fluorescence FITC gives some kind of okay, fluorescence isothiocyanate given but one kind of color. Then another that's color change. Another thing, the phycoerythrin, that is PE phycoerythrin, gives some color. These two things are recognized at a various stage, various steps, various wavelengths. Okay, this wavelength FITC recognizing that recognized at the wavelength of the 495 nanometer and 519 nanometer. Very very important thing about this FITC, the fluorescence molecule, green molecule, the FITC producing green color. That green color is absorbed under the 495 nanometer and 519 nanometer. It gives green color. Okay, this in this case of P, the phytochrome erythrin, this phycoerythrin is absorbed under the this wavelength called from 498 and 578, 496 and 578, which gives a red color. Its pigments of red color. Phycoerythrin, it recognize its remedy red color. FITC remedy green color. This is just explanation about this flow cytometry experiment. It's very important experiment in the any cell culture techniques, and any latest protein researchers are having the better results. And the last one test. Okay, another is a test. T is a test one, should be FITC positive. So here they mention there are 4 tubes. One is a control tube. This control tube is non-fluorescent label. So first one is non-fluorescent label. Second, that is the stand 1. That second stand having the S1 FITC labeled. Then second stand is the third stand is the phycoerythrin labeled. That is the third stand. Fourth tube which having the test control, that is the FITC positive. So 4 tubes are there. One is control, one is labeled with FITC, another one is labeled with the PE, and fourth one is labeled with the test. That test should be positive, FITC positive. So now the below facts results experiment, they are providing the experiment. This experiment, which is the correct one? Okay, they are providing the order. We want to select the one with the correct labeling. We want to find out the correct labeling. First one thing is control. C control is non-fluorescent label. Okay, non-fluorescent label. Many very important thing. They don't have any label. So yes is C. Very important thing. First thing, the control stand, there is no fluorescence. So it's in a zero mode. They not comes in this area. That is in zero mode. There is no labeling. Next come to this testings. Okay, what is the fluorescence labeled one? That S1. Okay, stand 1, it's a fluorescent label one. What is the fluorescent label? Here you can see this is the fluorescent label 1. This S1 is the fluorescent labeled one. That is the here no PE was not found. Here only the fluorescence alone found, FITC alone found. So they mentioned the S1. And another thing is the yes, they are S2. S2, that is only PE. That is phycoerythrin, only PE. And finally, the S2 here that test. Okay, this R should be maybe test or maybe S1 tube. That is the alteration one. Whatever we can take, we can take this two are in the same mode, so we can take both. So only they are mentioned the slash S1 R test, S1 R test. This is all about the fluorescence labeling. Very important concept. Just go through the Google or YouTube images. YouTube videos are available about the facts analysis. Just understand. Just flow cytometry result interpretation. Google it. Google it. Flow cytometry result interpretation. Result interpretation. Here the first column, that is no labeling. Here no FITC, no PE. Once you are that sample only having FACT labeling, you will get you will get the dots here. So that is the image R. This is the, that is only FITC labeling. Then the sample is labeled with only the phycoerythrin. PE means that comes like this. Here they provided the test. What is the test? Unknown sample test should FITC positive. So positive means a sample which having the FITC labeled molecule. So that image look like this. This are all about the facts. Okay, next. Okay, very easiest and interesting. And the most important experiment in the immunological method. In this immunological method, immunoprecipitation is very most widely used method. In this method, there are many kind of process are there. Immunoprecipitation in this method, that protein, okay, precipitating of protein antigen out of solution using the antibody. Okay, they are using the antibody in the experiment to accumulate to precipitate the antigens. Okay, the binding of antigen and antibody in the medium, that makes the accumulation, that makes the precipitation. This is the final output of this experiment. In this experiment, they are using the chromatin. In this chromatin precipitation, in the normally so normal experiment is the that is handled with only antigen and antibody. In this method, they are using the probe, DNA or protein interaction, DNA or protein interaction probe. These probes are they're adding the to the medium, and these probes are bound with the sample and they are getting precipitated. This helpful for the proteomic and genomic studies, and its main application of chromatin immunoprecipitation is the monitoring transcription regulation through the histone modification. Okay, very very important thing, monitoring the transcription regulation through histone modification. That is a epigenetic study. While they are doing a epigenetic study, we we want to select the chromatin immunoprecipitation assay. This chromatin, it is all about the chromatin immunoprecipitation assay. Whenever you are studying the content, if we want to understand the story, then only we will enter into the concept. Perform this assay, that is the chip assay, was performed to determine specific transcription factor binding site. Here they mention the transcription factor binding site on the promoter of the gene. Pull down was done using the either IgG or antibody against c-Myc, that is the protein against the antibody against the c-Myc. A DNA contains c-Myc binding region was used as a control for the PCR amplification. Which one of the following PCR presentation of the DNA is correct? Here the output, okay, they are run the PCR and collect the output sample of PCR, and they are loaded in a gel. Okay, this their assay was performed to determine the specific transcription factor binding site of promoter of gene. Now we want to know specific transcription binding site. They are using the IgG and IgG or antibody against mic. So these are all the outputs. In this output, you can see their input is the PCR sample. After the input addition, there is no binding happen in the IgG, some smaller amount, but the anti-C-Myc that are recognizing larger quantity of the that molecule, that's gene molecules, DNA strands. That but input is highly effective one, because in this they are having the DNA content that which having the uh, contains the c-Myc binding region. So in this experiment they are getting a better result with this anti c-Myc. Just imagine the story. First thing is they are doing the PCR. In that PCR input, okay, that PCR input having the higher mic binding domain. See the DNA containing the c-Myc binding region. Another case, this sample, okay, the after the PCR amplification, that PCR amplified samples are having particular, they are chromatin immunoprecipitating compounds, right? In this process the IgG is not recognizing the sample, because that not don't having the that mic binding region. But after the and this and addition, that well they are adding the anti c-Myc, they are receiving better resolution image, that having the better that bandwidth. Following certain statements regarding, next question, the following certain statements regarding somatic hybridization, that as I mentioned, first we are discussed the kind of hybridization. Here they are mentioning the somatic hybridization. This somatic hybridization method, we can see the larger that hybridization have a great potential in the plant improvement. The it is having the plant cell development. Okay, in the development of the hybrid plant through the fusion of somatic protoplast to different plants. This is called the somatic hybridization. This hybridization is this protoplast. Okay, this protoplast means it's known as the naked plant cells. That that component of plant cells excluding the cell wall. That all the looks like plant cell that don't having the plant cell wall. So these cells are hybridized with the fused with the two different plant species where another plant. So that plant is one that having the new character, that having the protoplast character. This is called the somatic hybridization. And this somatic hybridization is for example means that hybridization of for experiment they are doing hybridization of tomato and from potato. This tomato and potato hybridized plant is known as the pomato. Is one experimental value. Many of the plant researchers are doing this somatic hybridization. In this somatic hybridizations, many of the researchers are focusing because the upcoming the days, the plant production, agricultural playing a vital role in human evaluation, human revolution. So we want to do this kind of experiments. This is all about the somatic hybridization. In this technique used to plant improvement. Here they are. What is the correct combination? Q and R is the correct combination. Hybrids are produce a variable and asymmetric amount of genetic material of parental species. The another one is protoplast fusion pigments permits transfer of gene block and chromosomes. They are allowing the protoplast are allowing the fusion. Okay, that is the major region in the plant. Okay, at a major thing somatic. Whenever you are studying the somatics hybridization, the protoplast is a key role playing compound at having the similar organs like plant, though except the that cell wall. Next thing about this unit 13 in this question, part C question, nichrome coated stainless steel electrode. Nichrome coated stainless steel electrode were implanted in a rat brain for chronically recording the electrical activity. Chronically recording the electrical activity of deep brain structure. This is the statement. During the study of 3 months, the intensity of electrical signals gradually decreased. Why it's decreased? The following statement may explain the causes of the observation. Why it is decreased after during the 3 month study. Gradually, the electrical activity, that electrical signals are decreased. Why it is happen? The reason is P and Q. A deposition of metallic ions from the electrode tip cause the degeneration of some neurons. So that degenerates and promotes the degeneration of neurons as less electron signaling. Then gradual accumulation of microglia at electrode tip increased that resistance of electrode. Increases the resistance of electron. There is the two possibilities. One is degradation of metal ions and they are affecting neurons in the brain. So the neuron brain damage, it leads to the low electrical signal. Another reason, microglia are present, microbes which present in the brain that are accumulated on the electrode and they are increase the resistance of electrode. That microglials are that cells, okay, that microglial cells are resistance to the electrode, and then they stop the signals. Another point of question is number of seeds in the fruit. This is a very easiest and interesting question. It's a sum based question. Answer is I'm having in the like similar questions. I will send this telegram group, because this is the first question, like the first question you are see, right? Rejection and acceptance of null hypothesis. The number of seeds in the fruit of plant is H naught, that is the hypothesis. Mu is 30, the mean value, that standard deviation is the 30. The random samples are 9 random samples. Mean number of seeds as 24. Mean is 24. The standard deviation here is that sample is 30. Here the standard deviation for 9 samples is 6.12. First question, what are the confidence limit in the sample mean? What are the confidence limit in the sample mean means that is the that 24 plus 4, 24 minus 4, that is a constant confidence level. So that 24 minus 4, that is the 20, 24 plus 4 is 28. What is the rejection or acceptance null hypothesis at 95 percentage? This 95 percentage, that is a rejection of hypothesis. Rejection the hypothesis happen. Okay. Two homologous proteins were isolated from psychrophiles and thermophils. The psychrophiles and thermophils are the two kind of microbes which the present in that various region. This we all know this psychrophiles are present over the region called. Okay. This psychrophiles are this kind of on the bacteria. In this bacteria, they are living in the cold condition. Thermophile is they are living in the hot condition. This psychrophiles are can able to grow in a 20 degrees Celsius. Even at a 20 degrees Celsius, they can live in this particular temperature. And then another case about this psychrophiles is the extremophile microbes. Okay, extremophils that can grow in extreme conditions. Another thing about the thermophils are able to grow in a higher thermal temperatures. And then the purified protein, they had collected the protein from the extreme cold condition and extreme higher condition. Higher temperature means that is the 122 degrees Celsius, like till 121 degrees Celsius, because the the autoclaving temperature is 121. The microbe can grow at that 121 degree also. They are not able to decontaminatable. That is very highly temperature stable molecules, stable microbes. They are collecting the protein from the two microbes known as psychrophils and another is thermophils. The purified proteins are subjected to denaturation. Denaturation means what happened, protease digestion and circular dichromosome. Two type of degradation. One is protease digestion, another one is the circular dichromosome. The following observation was made. The CD is circular dichromosome, another one is the protein digestion. Okay, the inference is the what here they provide a four statement. The reason for enhancing the stability here, as per their four statement, CD spectra of P and T proteins are identical. CD and CD, this method using the protein digestion and protease digestion and this circular dichromosome shown similar protein degradation. Similar proteins are identical. They are mentioned. The proteins are similar. The amino acid composition is 95% identical. T and P are equally susceptible to proteolysis in the absence of reagent. T is higher midpoint than of thermal denaturation than P. These are all the statements given above, that enhancing the stability of T is due to the increases in number of salt bridges. Okay, while you are increasing the salt bridge, we can improve the stability of T. If they are improving the stalled bridges that can easily they are stable in a particular temperature. They are not brokenable. Next question is how to in an attempt to detect protein expression profile in a cell. Okay, they are doing the Western blotting. They are given a clue in an attempt to detect protein expression profile in a cell. Western blot technique is employed. Expression of two new proteins is to be followed by probing with respective high affinity antibodies. This is the respective high affinity antibodies. Unfortunately, the two proteins were found to co-migrate in a STS page profile. Under this situation, using one-dimensional STS page by Western blot, and they are doing the one-dimensional STS page and the Western blot, two experiments they are did. Which one of the following is the best way to demonstrate the presence of both protein? Here the story is given. They are asking the question which one of the following is the best demonstration the presence of both proteins. Once again I am repeat for they want to find out the protein profile. Expression of two new proteins. They have taken two new proteins. This is followed by probing in the high affinity antibodies. Okay, that's unfortunately the two proteins were found to co-migrate in a STS page profile. They can co-migrate in a STS profile. It's under situation one-dimensional STS page and Western blot. Which one of the best method is subjecting the technique of stripping and reprobing of gel and transferring to nitrocellulose membrane while doing the Western blotting. So this how to which one of the following is the best demonstration means demonstration is the how to impress, how to express, how to visualize. That is the one best method is the stripping and probing of gel after transferring to the nitrocellulose membrane while doing the Western blot. This is the right one. Okay, this is very easiest question and but big paragraph. You want to understand the story. Story start. A 6.4 kb plasmid DNA has two restriction endonuclease sites. Okay, this plasmid having the two restriction digestion site. That is the hint 3 and EcoR1. Complete double digestion of plasmid with the both enzymes yields the fragment 3.1 and 3.3 kb. After the complete double digestion means adding two enzymes. In order to study DNA repair process, G and T matching was introduced in one strand of hint 3 site. The damaged plasmid was incubated in a reconstituted repair system containing all factors and enzymes required for repair. If the efficiency of the repair system is 50%, which one of the following band pattern on the agarose gel will be obtained after treating of repaired plasmid cell with hint 3 carbon? Here, as per the question, this here the question is finished here. Okay, two fragments of 3.13 kb is obtained. This statement they are given for the confusion. Okay, in order to study there is no repair. Okay, they are doing a repair process. GNT mismatch was introduced. What induced at in the one strand of hint 3 binding site. After hint 3 binding site, that is the edge. Okay, that is not in the middle of the strand, so it won't influencing the that molecular weight. Damaged plasmid was incubated in a reconstituted repair system containing all the factors and enzymes required for the repair. If the efficiency of the repair system is 50%, this is the confusion statement. Okay, what we want to think, that is the confusion statement. So in which gives the following band pattern in the agarose gel. Which one is the after the treatment of both hint 3 and ECOR1? Here this in between statements are confusing statement. Here the question is finished at this stage and then continue here. The fragment of 3.1 and 3.3 kbs are obtained. What is the band pattern after the treatment of this both hint 3 and ECOR1? So the answer is D. They are obtaining the band, register band and the full length band. These are all the output. Okay, final tomorrow I will discuss the remaining. Okay, as per the schedule, day 18, day 19, day 20 is for the 13th unit discussion. Today is 13th. We are discussing some part of the 13th unit, the methods in biology. Day 19 and day 18 we will discuss some extra parts of both the unit 13, methods in biology. Thank you for listening. I'm in this session. If you have any queries, please post it and uh, post it in the Telegram group.